Welcome back to pharmacology, where we begin to look at toxicology. This is actually the broader study, and pharmacology comes under the banner of toxicology. So we are going to look at toxicodynamics and toxicokinetics. We'll apply a lot of these studies to drug investigations, where we'll look at how we might be taking a new lead, or it might be an old drug, a new development, and doing all of that risk characterization. Approving authorities that do this, including in Australia here, the Therapeutic Goods Administration, where they have to address such issues as are there at particular stages of the lifespan where that drug is more toxic. What happens in overdose situations with that particular drug. And we saw with drugs, absorption, distribution and excretion of the drug was important, and with drugs also their metabolism. So now when it comes to toxins, we'll just be more general. Toxins will include pharmaceuticals, herbs and a whole variety of poisons. So we've got to look at the ways that they are regulated and controlled. You have to, in your research, specify the context. You might be giving a regulatory authority in Australia or New Zealand or in an Asia Pacific nation, but you have to tell us which regulating authority for the use of that particular poison or toxin. Any particular lifespan issues. So of course toxicodynamics will be how the toxin works on the body, and then toxicokinetics will be how the body works on that toxin. We'll use general terms, so it will be elimination of toxins, it will be biotransformation. Metabolism is one example of a biotransformation process. So pharmacology comes under that banner of toxicology, looking at adverse drug reactions and drug interactions or drug-herb interactions and so on is part of that toxicokinetic and toxicodynamic assessment, where you do risk assessment, you look at overdose, you look at lifespan issues. We'll address age, pregnancy, lactation, childhood and elderly issue factors later when we look particularly at this situation of overdose. So the first concept that we hope comes out in your assignments is that toxicology is very broad. Here we've got a broad range of toxic agents. Some of them are chemicals, some of them aren't even chemicals. It's just any agent that is capable of producing a deleterious response in a biological system. So some of you will be researching perhaps Schedule 2, 3, 4 or Schedule 8 substances, or over-the-counter medicines, or it might be a complementary medicine. And these are capable of producing a beneficial response in a biological system, but they are also toxins. The principles are the same whether it's complementary medicine or Western pharmacy. Others of you might be very interested in particles that are released by chemicals such as alpha particles or beta particles, and if they release these particles they also more than likely release gamma radiation. So this is electromagnetic radiation. Some of you may research other forms of EMR, other energies, because they're agents that are capable of producing deleterious responses, and you'll research those in biological systems. But the majority of you will study xenobiotics, foreign organic chemicals that enter our biological system either in foods, in the herbs that we eat, with plant, animal or microbial origin toxins. You might be looking more out there in the environment, how we might become exposed in other ways, or it might be through our food webs and food chains and other medical aspects of toxicology. And this is the big category where most of you do your research. Schedule 5, Schedule 6 or Schedule 7 poisons, where you're looking at environmental or occupational exposure. So continuing this discussion of how very, very broad toxicology is. Here we have a list of chemicals. They range from inorganic to organic chemicals, and we're going to compare them for toxicity using one particular measure of toxicity called a lethal dose 50%, LD50. And this is one that you may come across if you do a substance that's used in a workplace. It may not even be a workplace. It might be more general. It might be the manufacture of the bottles or the packaging that are used for foods and so on. But because people will be exposed to levels of those substances, there will be a material safety data sheet available on that substance, an MSDS. And MSDSs commonly report this LD50. We're going to talk in some detail about what this LD50 means. Here's our deleterious agent. We're going to look at ethanol as one example. We're going to use ethanol quite a lot as our example substance to show you what we want you to do for your assignment. So it's a recreational drug, prone to abuse but not Schedule 9. It's quite legal. We're going to compare it with nicotine, also a recreational drug, not Schedule 9, quite legal. And you'll notice that ethanol has this LD50 of 10,000, whereas nicotine has an LD50 of 1. But now let's have a look at what LD actually means. It means the lethal dose, a way of measuring its toxicity. And when we say LD50, it's in 50% of animals exposed to that toxin. So we've got to control for other variables. They have different sizes. We've given their molecular formulas to show you an example of that. But we're going to give this substance in so many milligrams, but per kilogram of body mass of the animal system in which it was tested. And each MSDS will report which animal system was used, because that LD50 will vary depending upon the animals. It's just a way of getting an estimate of its toxicity or its risk. So to say that 10,000 milligrams of ethanol, when given as a dose per kilogram of body mass, is lethal to 50% of the population, whereas nicotine, only 1 milligram per kilogram of the body mass of that test animal used, would kill 50% of the population, then nicotine is more toxic. Only takes 1 milligram to kill half the population. So the substances below nicotine are the more toxic, right through to the very deadly. But also note that there are some inorganic substances there. One that we use as a food substance, any chemical in excess amounts beyond normal exposure levels has a toxicity profile. So the ones below nicotine, various plant and animal toxins, some industrial substances, and then last of all, botulinum toxin, one of the most toxic substances known. It only takes 0.00001 milligram per kilogram of the body mass of the test organism to kill half that population. Now, diluted 1 in 1,000, it is actually therapeutic. People who have terrible strictures can then regain some use of the structures at that joint, so that musculoskeletal therapy or physiotherapy can then recover use of the muscles after some degenerating neurological illness. And also, diluted to that 1 in 1,000, that toxin is also known as Botox, so it has cosmetic value as well. And so here we have a substance that you might want to research, but you wouldn't research its cosmetic or its medicinal uses, though you would mention those as important sociological impacts, but you would research and then report on the toxicity profile. But more on that LD50 shortly. If you don't think you've got the concept yet, we're going to explore it later when we do dose estimates of toxicity. One of the major principles, though, that we're getting out here is that toxicity depends on dose. You take a substance or are exposed to it in the environment, and that particular dose will lead to a measurable response, but we'll see with toxicology that time is also a critical parameter. But other factors to consider here, it's not just straight dose response curve, so many impacting variables. We're going to see that volume of distribution, which tissues are involved. We'll look in more detail at those shortly. But as to how toxic a substance is, it does also depend upon the route or the site by which you were exposed to it, and, as we've hinted, the timing of that exposure. So all of this is put together by the regulating authorities and done as this risk characterisation. So you're going to perhaps mention some of the legislative controls that you may have become aware of, or the regulatory controls over the particular toxin or toxins that you'll research. Now, one of the other reasons why we've got to do this toxicology study was for these idiosyncratic reactions. We want some population data so we can say at least 10% of people might show this bizarre allergic reaction to this particular toxin. Now, the timing aspects. Your exposure could be acute, could be subacute, could be subchronic, or could be a chronic exposure. If it's an acute exposure, we're after acute toxicity measures, and the worst of all for a toxin would be acute lethality, where we've said the way of getting a measure of that acute lethality is that LD50 study needed for those material safety data sheets, MSDSs, a wonderful source of toxicology parameters on some of your industrial poisons. But if it's a subacute exposure, a repeated dose, say washing your hair, using that substance in a workplace as a skin antiseptic, you want to know if it has any subacute toxicity, where you would have to then do skin sensitization studies. We'll talk more about those skin and eye irritation studies later when we look at ways of reducing animal deaths in these toxicology studies. For some substances, there might be a subchronic exposure pattern, where you then have to test the substance for its toxicity, but using repeated doses. And we'll see that one of the measures of safety there, a way of estimating the risks with this toxin, is a LOAEL, an LOAEL, a lowest observed adverse effect level, but more on that shortly. Then again, your exposure to that toxin might be chronic. It might lead to changes such as oncogenicity, a mechanism of toxicity that we went into some detail exploring, so that you would know the level of detail that you would need to go to in your assignments. You wouldn't just mention that a toxin was oncogenic. You would try and find out which proto-oncogenes it affected. And because it has this chronic toxicity pattern, some of these other studies, such as an LD50 or a LOL, might be unimportant. This other one that we're going to talk about, a TD50, what you may need to find for that particular substance, because it has a chronic toxicity profile, is its maximum tolerable dose. So the toxicology parameters you report will depend upon your particular toxin. But with all of them, that dose response principle will be the same, but we want to measure what is the safety margin with that dose, what's the range. So we'll need to know some toxicokinetic parameters such as clearance, volume of distribution and half-life, but more on those shortly, just as a review. Further to reviewing a couple of terms that we've looked at, we were interested in establishing for substances that are therapeutic what is a good therapeutic range with minimal toxicity. And we used an effective concentration 50%. In fact, we saw that the therapeutic range often used anywhere between EC20 and EC80, but this percentage, 50%, 80%, was a percentage of a maximal response that drug shows in those tissues. Now, what we're talking about is population studies, and so when we say something is an ED50, that's in 50% of that population. And so, to interpret a toxic dose 50%, that dose that was toxic to 50% of that population. We'll go into more detail shortly and look at some statistical controls that are placed there. So ED50 is effective dose in 50% of the population. Now that might be relevant to some of your toxins because they have a therapeutic effect, but for most of you, your toxins will not. So that won't be the parameter that you'll research. But that ratio of TD50 divided by ED50, we're going to come back and talk about that one because that's that therapeutic index, a concept that we're going to refer to quite regularly when we look at drugs affecting the body's systems. So it's important to realise that that's brought out from population data, and it is found from looking at those dose response curves. And so some of these other toxicokinetic parameters that we need to review. Clearance, first of all, we use that in helping work out what would be a good maintenance dose for a drug. So when we're talking about non-drugs, when we're just talking about xenobiotics in general, foreign chemicals that have come from one biological system into our biological system, we're going to need to clear that toxin from all compartments of the body, or we're going to need to know in which compartments that toxin sequesters or becomes hidden away. With volume of distribution, we needed to know that particular theoretical calculation for a given drug when doing a new drug investigation on it. That helped work out a loading dose. But similarly with studying toxins, xenobiotics and other chemicals that are non-drug, we've got to know that volume of distribution. It's a theoretical calculation, but with toxins it often involves more than one compartment. We'll see once again, because of this sequestering in a particular compartment, that volume of distribution has great impact on how we detoxify these environmental and other toxins. Now, once again, with drugs, half-life was important because we needed that to know a dosing interval. Were we going to take this drug once a day, or was it needed twice or more in the one day? With xenobiotics, non-drug substances, we also need to know the half-life. It's a constant for that particular foreign toxin, and we need to know how many half-lives there would be sufficient to clear that toxin from the body. And as we'll see, you can never really get some toxins back to zero levels, but where we want to get them back to an acceptable margin of safety. But more on those margins of safety shortly. All of this data comes out of dose-response curves. This fundamental concept that the higher the dose, the more severe will be the response to a toxin, and you should be able to look at the effects that you see in that person and correlate that they were exposed to a particular dose and report on that in the literature to help once again with that management in the future. So population data, always here. It's not just 50% of a maximal response shown in that tissue. This is in a whole variety of in vitro cell lines or tissues from animals or whole animal studies. And with some drugs we can study them and work out dose response curves using human clinical subjects. So working on this idea that a particular dose gives a measurable response. We can use this then as a way of estimating and predicting risk or effects from toxins that might be induced. if it's a substance that is therapeutic, we can work out an effective dose. That's a dose estimate of its effectiveness. And we can likely also work out an LD50, a lethal dose. That's another dose estimate of the toxic effects, where we've said an example is an LD50, one that is regularly reported in MSDSs. But that's an acute toxicity lethality. A lot of toxins, the problem is that they are still toxic in doses much, much lower than their lethal dose. Also, that LD50, it was a statistically derived parameter, and where we're looking at a population once again, and that 50% of the individuals in that population would be expected to die at that particular dose. But oftentimes we need to know toxic effects, so there would be a TD, a toxic dose, as a dose estimate of toxicity. To show you the principle then of how you work those toxicokinetic parameters out from a dose-response curve, toxicodynamic properties from a dose-response curve, we have a population of subjects, animals usually, with an LD50, because it's morally and ethically possible to do it with animals if you will apply to various animal ethics committees. Now, you actually interpolate the LD50 from your available data points, and you can see the little tags. We've tested many, many animals within that population, and that's the range of responses in those populations at repeat measurements, and we're looking at an average or a median, a curve of best fit, and once again you can see that sigmoid curve. We've done that semi-logarithmic calculation on it, well, during graphing. So the LD50 interpolated in this particular case by reading across to the graph at the 50% of the population, read back down to, we predict that it would take a dose of 20 milligrams of that substance to kill 50% of the individuals in that population. That would be reported as the LD50. You won't see the dose response curve reported, just that parameter as a measure of the toxicity. But that may be unacceptable for some toxins. We may want to know a dose at which you would expect no individual to die. So that would be an LD0. That's a just below threshold for that particular response where we're looking at lethality. So there are other measures. There might be an LD10, more on that shortly, but as you can see, the dose at which you'd expect 10% of the individuals in the population would die. Now that C that you see here in LC50 is for inhaled toxins, where you're looking at air concentrations, and not to be confused then with EC50, which was an effective concentration 50%, where that 50% referred to 50% of a maximal response. No, this is LC50, a concentration expected to kill 50% of a population. So there's others, LC0, LC10, and so on. We'll talk about some of those shortly. So here is one parameter that is only relevant if this toxin has therapeutic effect. So we've given an example to help you understand this concept. It produces relief of pain, this particular toxin that we're studying. However, as dose increases, there is also some harmful changes going on in those tissues, leading to an endpoint, something readily observable. In this case, the harmful effect seen is paralysis. So the ED10, the effective dose, where 10% of the population showed relief of pain. That would be compared to a control population for statistical purposes. And ED50 was the dose that was effective for 50% of the population, causing pain relief for those 50%. And this was once again compared to a control. ED90, a dose effective for 90% of the population, producing pain relief. So the more you increase the effective dose, the greater the chance then of that dose producing a toxic response. In our particular case, that was paralysis. So it's still a dose estimate of toxicity, where a TD0 is that dose that is toxic to 0% of the population. TD10, 10% of the population showed the paralysis, compared to the control. TD50, 50% of the population showed paralysis, but remember, that may not have been a data point that was measured. That may have been interpolated from the dose-response curve. And then lastly, TD90, the dose that is toxic to 90% of the population. Here, to study toxicity and to save animals, we may use animal cells or human cells and scale it up, predict for living human systems. So we've given some examples there of how you could do a TD, where this time you wouldn't be looking for paralysis, a whole of body effect. You'd look for one of those earlier cellular changes and report and measure on that. So some examples: epidermal keratinocytes or corneal epithelial cells might be your cell model that you study, and then you predict the TD50 for changes to the skin or eye of a living system. So the cell line is in vitro, and scaling it up to the whole animal or person is then in vivo. So to show you that then on a dose-response curve, here we have a substance that has this therapeutic effect. It produces pain relief. There are some sub-threshold levels of doses of that drug or herbal medicine. At the effective dose 50%, note it's causing that pain relief in 50% of the population. That's different from the EC50, and you can see then that at the ED50, when you look at how much toxicity you see in the population, that's a TD0. None of the population shows the paralysis, that toxic effect, though half the population was already starting to show the beneficial effect of the pain relief. And we ask you to look at those curves and note that even up to about an ED 75%, there is no toxicity pattern as yet. However, then on the curves we want you to look at the ED 90, where you're just increasing the dose of that substance till you get pain relief in a larger number of the population, this time now 90%. When you scale that back down here, interpolate to the dose response curve for the toxic response, you begin to see we now have a toxic dose. So we ask you to estimate what that might be by looking at where the ED90 crosses the purple toxic dose response curve and read it back to the vertical axis. So we estimate a toxicity of about a TD1 or a TD2. 1% or 2% of that population will be starting to show the paralysis, the toxic effect at a dose that leads to pain relief in 90% of that population. So NOL and LOL are other measures that are used to estimate margins of safety with particular toxins. So they're threshold measures where you just see an effect, the lowest observed adverse effect level, or it might be something that just disappears from the population. The difference between a NOL and a LOL is merely mathematical and statistical, and we'll see that when we look at a dose response curve shortly. When we say that there is no observed adverse effect level, you'll see that this doesn't mean that there was no toxicity seen. Some members of the population could have shown our toxic effect, which was paralysis, but that's not statistically significant when compared to the control group, and so it's only when you get that result that could not have happened by sheer random chance, statistically significant difference from the normal control population, that's when we have the lowest data point that is statistically significant, but where you observe that adverse effect level. So showing it on a graph, there we begin to see some adverse effect in the population. The percentage that show that response, let's say it is that paralysis, is there, but it's not statistically significant. So the next measured data point, that particular level, that particular dose, that's the LOL, the statistically significant one. Okay, so once again, worked out from that population dose response curve. Now, you don't often see the dose response curves in the literature. Don't break your heart over trying to find them. That's raw data. Often all you will find reported is the processed parameter, that one measure that summarizes the toxicity profile of that substance. So this was one of those calculations that can be done on that raw data. That was that therapeutic index done for drugs like digoxin, lithium and others. Digoxin and lithium having this low therapeutic index. Well, let's have a look at what that means. So therapeutic index was a ratio of two dose estimates. We have the effective dose, because this substance has a therapeutic effect, and then we have the toxic dose. Or we could use an LD, comparing it then to the ED. But the ED is always the divisor, it's always the denominator in a ratio. And so it's not always the median data points that are used, but as our example, we have used the TD50, which is 200 milligrams, and an ED50 of 20 milligrams. Note the dose where it starts to show the beneficial effect, which was our pain relief, 20 milligrams of that substance. Pain relief in 50% of the population, though. But at 200 milligrams, that was where we got a toxic effect, paralysis, in 50% of the population, and we divide those two numbers to come up with a therapeutic index of 10. Digoxin and lithium commonly have therapeutic indexes of 3. The fact that that's a lower answer when you do that division tells you that the toxic doses are too close to the effective doses. So it's that measuring how far different from toxicity, how far from toxicity are we? And the same will be important when you look at environmental poisons, where they don't have any therapeutic or beneficial effect. So a TD or an ED will be irrelevant to them. The measure that is done on a dose response curve is that NOL or LOL. But the NOL is often used because it's erring more on the side of caution, because here you're looking for a subtraction. You have a human population that through an industrial accident or something like that was exposed to a particular estimate dose based on their responses, their level of consciousness and so on, or some actual measurement that might have been taken after the event, after the disaster. And so you compare that to the NOL, subtract them to get a margin of safety measure. So sometimes margins of safety are reported for drugs, and once again you don't want a small margin of safety. So something with a margin of safety less than 1 would be are used with caution when prescribing that drug. You'd want a margin of safety greater than 1 to be assured that your effective doses were quite far away from your toxic doses. Now, some other important considerations with whether you'll find a TD or an LD reported for your particular toxin. we did indicate that if it's in the workplace that you could be exposed, there will very likely be an LD50 available on that toxin on its MSDS, its Material Safety Data Sheet. Animals had to die to get that value, but once it is known and reported by a primary source, all of these secondary sources, whatever the company that manufactures that substance and puts out that MSDS, will give the same LD50, or if it's a different one, they'll specify which animal system it was studied in, but it's always some animal has to die. You've got to get ethics approval for that. There are still ways of minimising the number of animals that have to die for procedures that will never benefit an animal, but only a human. So there's no substitute for that LD50. There's no in vitro or in silico substitute, whereas we saw for the toxic dose where it might be skin sensitization or ocular toxicity, we can use cell lines. There are substitutes. There are some substances where you will actually find a dose response curve, and it's probably because it's so unusual. It's not the usual sigmoid shape. So having said, don't break your hearts trying to find a dose response curve. You may not. You may have just picked the right idea for your toxin, where there will be this U-shaped dose response curve, which is known as a hormetic dose response curve. And you're going to see that if you've chosen an essential nutrient to study for its toxicology profile. We'll look at some other examples of substances that produce this U-shaped curve, and we've chosen a vitamin. Could be a trace element, but what we're saying is at 0 dose, that would also cause death to that person, and at certain doses, serious pathology leading to death. When you hit the homeostatic range, the person could still be taking it at low levels, but within recommended amounts, their body mechanisms able to compensate and repair, so normal homeostasis. Then at the other end, they're taking that nutrient in too high amounts, but not as yet producing serious pathology, where once again homeostasis, the body is able to compensate for those toxicity patterns that are starting to appear in the cells and tissues. But eventually, if doses increase, then dose response principles kick in. You will eventually see the endpoint toxic response. So for vitamin C, it has a particular toxicology profile. It produces diarrhea. Vitamin A is neurotoxic, and that's quite an acute toxicity too. You can be acutely overdosed on vitamin A, so you may wish to research those substances not for their therapeutic and nutritional benefits, but for their toxicology profile. There are other micronutrients where a 0 dose is toxic to you and where there's a homeostatic range, but where high doses are toxic to the population. Not necessarily vitamins, we said trace elements. You can go ahead and maybe follow through with that idea if you haven't yet picked a toxin to study. So they produce this U-shaped hormetic dose response curve. Low doses, mildly protective, no dose toxic, high dose, perhaps some long-term chronic effect or perhaps an acute effect. We're going to use alcohol as our example to show you what we want you to report in the literature. If you do find a curve, well then of course try and duplicate it, but as always, diagrams you compile yourself are always given more value at the integrated, comprehensive or standard level than ones you just merely copy and paste electronically. Alcohol showing this J-shaped dose response curve at very, very high doses, we'll see clearly quite toxic. At mild doses though, it's actually deemed to have some beneficial effects, because using more retrospective data than any actual study, it is predicted, it's a theoretical no dose could be toxic in the population. Problem with alcohol, as we'll see later when we look at it in a bit more detail, it's metabolized so as to detoxify it. But during that process, it does remove some of the hydrogen atoms and stores them on our important coenzymes that are made from vitamins, and we can then use electron transport chain to turn those hydrogen atoms into, well, their energy is turned into ATP, or stored in ATP to be technically correct. So because alcohol has this calorie value to it, chronic alcoholics don't want to eat, and that produces a different toxicity profile in them, as we'll see. So there's alcohol's J-shaped dose-response curve, our hormesis, where certain doses may actually be effective, but very, very low doses, very, very quickly, as you increase the dose, you start to hit the toxic effects, where it's more of a J shape instead of a nice gentle U. The toxic leading through to the lethal effects occur at quite a rapid rate as you increase dose. So the toxic dose that you choose might not be death, it might be fatty liver that appears, or cirrhosis, or esophageal cancer, but as you'll appreciate, death is a sequelae to those, and the higher the dose, the severer the response, and so the earlier the death. However, at these low doses where it is protective, the adverse effects that are not seen in the population, so something that disappears from the population, certain coronary diseases, and cerebrovascular problems such as a cerebrovascular accident, also known as a stroke. And so it's predicted then that with no dose of that alcohol in the population, there would be a greater incidence of those adverse effects in the population and a greater risk of death, but not the rapidly increasing J shape, the gentle part of the J there at the 0 level of alcohol in the population. So in summary, we said once you've chosen your toxin, you're not going to find all of those data points on it. Some of them are not relevant to your toxin because it has no therapeutic effects. Others of them are environmental toxins, where a NOL or a LOL might be more important. And these were ways of doing an estimate of safety using a margin of safety measure to estimate the risk. Whereas if it had some sort of beneficial effect, we might do a median ratio, a therapeutic index, a TD50 divided by an ED50. There were other ratios. It didn't always have to be medians that were used, and here we've given the example of an LD1 over ED99. The dose lethal to 1% of the population. Divide that by the dose that is effective in 99% of the population, and look at that ratio as a therapeutic index, giving us a margin of safety. Having said that you won't find dose-response curves, you will only find reports of those processed data points from the curve. There are some curves that are just so unusual you may just find them reported in the literature, and those will be those hormetic dose-response curves. We stress that with these dose-response curves that all measures are done in population studies. They are compared to a control group that is not exposed to that toxin, that may have some sort of placebo procedure or substance used on them. And then we look for a statistically significant change from those two population groups that could not just have happened by sheer random chance. The LOL, the statistically significant toxic effect seen in a population, whereas the NOL, often used where we want to be very, very cautious, err on the side of caution with doing a dose estimate of toxicity. And so that was the first time that we have a measure where we saw a toxic effect in a population, whether it was statistically significant or not.